Hello everyone. In this lecture, I'm going to discuss about adaptive immunity. So this will be just an overview and the cells that are involved in adaptive immunity. The first point you have to remember is lymphocytes. Lymphocytes, these are the predominant types of cells that are involved in adaptive immunity. It can be divided into humoral immunity and cell-mediated immunity. So humoral immunity is against the extracellular microbes and their products. And cell-mediated immunity is against the intracellular microbes and cancer cells. Okay. Adaptive immunity can be divided into humoral immunity and cell-mediated immunity. Humoral immunity is against the extracellular microbes and their products. Cell-mediated immunity is against the intracellular microbes and cancer cells. Humoral immunity, it is mediated by the B lymphocytes. It is mediated by the B lymphocytes and their products, that is antibodies. So B lymphocytes, when they are stimulated with antigens, they will be converted to the plasma cells, and the plasma cells will produce antibodies against the antigens. Okay, so B lymphocytes and their product, that is antibodies, they play a role in humoral immunity, and T lymphocytes, they play a role in cell-mediated immunity. We'll discuss in detail about all these things in our subsequent lectures. Now we'll discuss some basics about this B and T lymphocytes, and we'll also discuss about some other cells of adaptive immunity. Now let us discuss about lymphocyte diversity. So what is this concept of lymphocyte diversity? Again, this we have discussed when I discussed about the thymus and T cell development. Okay, actually, whatever we are going to discuss in this lecture, most of the concepts I have already discussed in my previous lectures. Okay, so as we have understood all those lectures, I mean, as we have studied about the lymph nodes, the spleens, the thymus and T cell development, we have studied the normal hematopoiesis and the bone marrow. Because we have already studied all those things, this lecture is going to be very easy. It's just a combination of all those concepts. So what do you understand by lymphocyte diversity? Lymphocyte diversity means lymphocytes in our body, they are programmed in such a way that they can identify various types of antigens. That is possible because the lymphocytes have different types of lymphocyte receptors. Okay, so if you see, this is a lymphocyte, it can have a receptor. It can have receptor to identify a particular antigen like this. They may have a receptor like this, which can identify another type of antigen like this. Or they may have a receptor like this, which can identify another type of antigen. Okay, so lymphocytes can have different types of, different lymphocytes can have different types of receptors to identify different types of antigen. But one point you have to remember that one lymphocyte will have only one type of antigen receptor. Okay, this diagram is just to simplify the things, but you have to remember one particular lymphocyte is going to have only one particular antigen receptor. That means one lymphocyte is going to identify only one type of antigen. Okay, but different lymphocytes will have different antigen receptors so that they can identify different antigens. Now, how is this possible? Okay, how it is possible to make different receptors for different lymphocytes? That is because of the VDJ recombination that is happening at the genetic level. Okay, so we have already discussed that the genes for the lymphocyte receptors, they are present as gene clusters in the chromosome. Okay, so if you see here, these gene clusters of the lymphocyte receptors, this can be named as V, J, C, and D. V means variable, C means constant, J means joining, and D means diversity. Okay, so if you see the genes of lymphocyte receptors, these are actually present as gene clusters in the chromosome. Okay, and these are named as V, J, C, and D. And these gene clusters, they are spatially separated. They are spatially separated in the chromosomes. That means these gene clusters or a particular DNA segment inside this gene cluster, it cannot be transcribed independently. Okay, if you take a particular segment, a small segment, small DNA segment of inside this gene cluster of lymphocyte receptor, this DNA segment independently, it cannot be transcribed. That means this DNA segment cannot be expressed. This gene cannot be expressed. Okay. Now, how they can be expressed? How these gene segments can produce the lymphocyte receptor? That is possible only because of a gene that is known as RAG gene. It has two products, RAG1 and RAG2. They are going to activate another protein, or you can say another enzyme that is known as VDJ DNA recombinase. VDJ DNA recombinase. So what is the function of this enzyme? The function of VDJ recombinase is to cut the DNA segments. Cut the DNA segments in these gene clusters of lymphocyte receptors and ligate multiple segments in random fashion. Okay, so this enzyme VDJ DNA recombinase, it is going to cut the DNA segments that are there in the gene clusters of lymphocyte receptor into multiple small segments, and then it will cause ligation of those DNA segments in a random fashion to produce multiple types of DNA segments. Okay, so they can cut DNA and ligate in random fashion so that different types of DNA segments can be formed, and now these DNA segments can be transcribed and they can produce various lymphocyte receptors, and this is true for both B lymphocyte and T lymphocyte. That means they can produce variety of B cell receptors in the B lymphocytes and variety of T cell receptors in the T lymphocytes. Okay, so I hope this concept is clear. This is how lymphocyte diversity happens. Okay, because of this VDJ DNA recombination, the lymphocytes develop different types of antigen receptors and they can identify different types of antigens. Okay, one more point you have to remember is this gene clusters of lymphocyte receptors, they are present in all the cells in our body, not only in the lymphocytes, they are present in all the cells in our body. However, this RAG gene is not activated in other cells. These are activated only in the lymphocytes. That is why lymphocytes will express, they will express the lymphocyte receptors. Okay, they can only produce the lymphocyte receptors. Other cells in our body, they cannot produce lymphocyte receptors because they do not have RAG gene. If they do not have RAG gene, they cannot make this recombination of various gene clusters. Okay, so this is the basics of lymphocyte diversity. So if you see for an example, you can see these are the lymphocytes. These are various lymphocytes, and you can see some of the lymphocytes have a particular type of antigen receptor. This is one group of antigen receptor. Then you can also identify that there is another group of antigen receptors. And also there is a third type of antigen receptors like this. Okay, so this is known as the lymphocyte diversity. That means in our body, various lymphocytes have a particular type of antigen receptor for a particular antigen, and they are different from each other. Okay, once a particular type of antigen is exposed, once the body encounters a particular type of antigen, only those lymphocytes which have receptor for that particular antigen, those lymphocytes will get activated. Okay, so for example, if this antigen is exposed, only those lymphocytes which have receptor for this antigen, only those lymphocytes will be activated. Okay, so upon antigen exposure, only those lymphocytes that have receptor for this particular antigen, they are selected. They are activated. And this process is known as the process of clonal selection. This is known as the process of clonal selection. That means the clone of lymphocytes that have receptor for that particular antigen is selected. That is known as the process of clonal selection. Okay. Now these lymphocytes will activate. These lymphocytes will be activated, and they will produce the immune response against that particular antigen. Okay. So if you see the numbers in our body, we have around 10 to the power 12 lymphocytes. And if you see the clones, we have around 10 to the power 7 to 10 to the power 9 different types of lymphocyte clones. Okay. So what do we mean by clones? Clones mean a particular clone will have antigen receptor against a particular antigen. That is known as one single clone. We have around 10 to the power 12 lymphocytes, and if you segregate them into clones, we have around 10 to the power 7 to 10 to the power 9 clones. Then what is the concept of polyclonal and monoclonal? For example, if the patient has a particular infection, during infection, the microbe will produce different types of antigen. The microbe is going to produce different type of antigen. Now, this will cause activation of different types of lymphocytes. Okay, that means different clones of lymphocytes will be activated whenever there is an infection. Okay, here I'm just giving an example. You can say any non-neoplastic immune response, is going to produce this polyclonal type of lymphocyte activation. However, neoplastic lymphocyte proliferation, whenever there is neoplastic lymphocyte proliferation, it is going to activate only a particular clone of lymphocytes. Okay, so this is how we differentiate. Whenever there is lymphocytosis, we can actually differentiate whether it is a non-neoplastic or it is a neoplastic condition. Okay, so if you put markers to see whether the lymphocyte belong to a single clone, and if they actually belong to a single clone, that is known as monoclonal lymphocytosis, and that implies a neoplastic condition. But if the markers are not identifying a particular clone, we tell them as polyclonal lymphocytosis, and that is usually found in non-neoplastic condition. Okay, and because they have different types of different types of antigen receptors, we can actually use markers to identify different clones of lymphocytes. Okay, so this is the concept of lymphocyte diversity, clonal selection, and polyclonal versus monoclonal lymphocytosis concept. Now we'll discuss a little bit about the T lymphocytes. Again, this I have already discussed when I discussed about the thymus and T cell development. So basically, I want you to focus on the T cell receptor. Based on the T cell receptor, they are divided into alpha beta type of T cells. These constitute around 95% of the T lymphocytes in our circulation. We have another types of T lymphocytes like gamma delta T lymphocytes and NK T cell T lymphocyte. So these constitute around 5%. So 95% is alpha beta type, 5% can be gamma delta or NK bar T cells. Now, if you see the structure of alpha beta T lymphocytes, but before that, you have to remember that T lymphocytes, they constitute around 60 to 70% of the all peripheral blood lymphocytes. Okay, so this point is also important. This number is also important. So in the peripheral circulation, if you take all the lymphocytes, around 60 to 70% are T lymphocytes. Among those 60 to 70% T lymphocytes, 95% have alpha beta type of receptor, 5% can have either gamma delta or NK T cell receptor. Okay, and if you see the structure of alpha beta receptor, it has alpha beta dimer, as you can see here, alpha beta dimer, and it also has associated proteins to make the complete T cell receptor complex. What are the associated proteins? These are CD3 proteins. These are CD3 proteins and the eta chains. These are the eta chains. Okay, so this epsilon gamma, this is one CD3 protein. Delta epsilon is another CD3 protein. Like that, there will be multiple CD3 proteins, and also there will be eta chains. So alpha beta dimer will be attached to CD3 proteins and eta chains, and they will complete the structure of T cell receptor complex. Okay, and also they will have various co-receptors like CD8. Some of the T cells can also have CD4, and they will also have CD28. Remember, all the T lymphocytes, they will have CD28, but they will have either CD8 or CD4. Okay, none of the T lymphocytes in the peripheral circulation is going to have both CD4 and CD8. Okay, so if the T lymphocyte has both CD4 and CD8, that is known as double positive T lymphocyte. That is only found in the cortex of thymus. Okay, so that we have already discussed when I discussed about the thymus. In the peripheral circulation, the alpha beta T cell receptors, they will have the alpha beta receptor and they will have co-receptors. This co-receptor can be, this co-receptor will include the CD28 and also it can be CD8 or CD4, not both CD8 and CD4. Okay. So this is a structure of CD8 positive T cell, that is cytotoxic T cell. Okay, because it is having the alpha beta receptor, along with that it is having CD8 receptor. You can say CD8 co-receptor. And we have discussed that antigen presenting cell or any kind of cell which is expressing the MHC class 1 molecule, and if the cell presents antigen with the help of MHC class 1 molecule, this can be identified by the CD8 plus T cells. Okay. And if you differentiate the CD4 and CD8 T cells, CD4 and CD8 T cells, out of this 95% of alpha beta T cells, CD4 T cells are around 60% and CD8 positive T cells are around 30 to 35%. Okay. So 95% of the T cells in the peripheral circulation are alpha beta receptors. Among these, CD4 positive alpha beta type of T cell receptor are 60% and CD8 positive are 30%. And one more next point is this alpha beta type of T cells, they can only identify peptide antigens. They can only identify peptide antigens presented by the MHC molecules. Okay, now we'll see a little bit about the gamma delta T cells. So basically, gamma delta T cells, they do not need MHC. They do not need the help of MHC molecules to identify the antigens. They can identify the peptide antigens, but along with peptide antigens, they can also identify lipid antigens and small molecules. Small molecules, they are present in the epithelia of the skin, and also the mucosal surfaces of genitourinary tract and gastrointestinal tract. Okay, so gamma delta type of T cells, they do not need MHC molecule to identify the antigens. They can identify peptide antigens like the alpha beta T cells, but along with that, they can also identify lipid antigens and small molecules. And they are present in the epithelia of skin and the mucosal surface of gastrointestinal tract and genitourinary tract. If you see the NK T cells, again, these are not same as NK cells. NK cells are different. NK cells play a major role in the innate immunity that we'll discuss under the innate immunity section. NK T cells, they do not need MHC molecules, but they need MHC-like molecule, which is known as the CD1. Okay, so they need antigen presentation with the help of CD1 molecule, which is a MHC-like molecule, and they identify glycolipid antigens. Okay, so alpha beta T cells, they can identify peptide antigens presented through MHC molecules. Gamma delta T cells, they can identify peptide antigens, lipid antigens, and small molecules without the help of MHC molecules. NK T cells, they can identify the antigens, especially the glycolipid antigens, with the help of a MHC-like molecule, which is known as the CD1. And of course, the numbers are important, as I have already told you. The numbers are important. T lymphocyte, it constitutes around 60 to 70% of the peripheral blood lymphocytes, out of which 95% are alpha beta type and 5% are, it may be gamma delta or NK T cell. Okay, and among these alpha beta T cells, 60% are CD4 positive and 30% are CD8 positive. So these are the basics of T lymphocytes. Now we'll discuss a little bit about the B lymphocytes. So B lymphocytes, they constitute around 10 to 20% of the peripheral blood lymphocytes. Okay, so T lymphocytes are 60 to 70%, B lymphocytes are 10 to 20%, and around 10%, 10 to 15%, you can say they are NK cells. Now we'll see what is the structure of B cell receptors. So basically, the B cell receptors are nothing but immunoglobulins. Okay, so if you see the B cell receptor, it is nothing but immunoglobulins of class either IgM or IgD. Most of the times it is IgM. Okay, so B cell receptors are nothing but the immunoglobulin molecules, either IgM or IgD, but most of the times it is IgM. Okay, so if you see here, this is the immunoglobulin molecule. This is the immunoglobulin molecule. Okay, but along with this immunoglobulin molecule, there will be certain other receptor to complete the B cell receptor complex. Similarly, what we have discussed for T cell receptor, along with the alpha beta dimer, there will be other associated proteins which will make the entire T cell receptor complex. Similarly, for B cells, along with the immunoglobulin molecules, there will be some associated proteins which will complete the structure of B cell receptor complex. So, what are these associated proteins? These are known as immunoglobulin alpha and immunoglobulin beta. So, these are immunoglobulin alpha and immunoglobulin beta. Okay, otherwise known as CD79A and CD79B. Okay, of course, the B cell receptors will also be diverse, and the diversity is caused by, it is also diverse, and that diversity is caused by the recombination that we have already discussed with the help of RAG gene. Okay, so the B cell receptor complex is very simple. It will have immunoglobulin molecule, IgM or IgD. Along with that, it will have associated proteins like immunoglobulin alpha and beta, otherwise known as CD79A and CD79B. So this is the B cell receptor complex. Now, what are the co-receptors that the B cells can have? So if you see the co-receptor, I mean, for T cells, we have discussed the co-receptors are CD28 and CD8 or CD4. For B cells, the co-receptors include, this is actually a co-receptor. It is a complement receptor basically. It is known as CR2 or CD21. CR2 is complement receptor 2 or otherwise known as CD21. It's very important because this is also a receptor for Epstein-Barr virus. This is also a receptor for Epstein-Barr virus. Okay, so Epstein-Barr virus, they have predilection to infect or involve the B lymphocytes. And that is possible because of this receptor, this co-receptor. Okay, and there is another co-receptor that is known as CD40 receptor. So this is the CD40 receptor. Okay, so what are the co-receptors? Co-receptors. So co-receptors include one complement receptor, which is known as CR2 or CD21. Another co-receptor is CD40. Okay, so if you see this B cell receptors, they can identify the antigens. Some of these antigens will be opsonized with the help of complement proteins. Some of these antigens will be opsonized with the help of complement proteins like C3b. And those C3b can be identified by these complement receptors. Okay, so primarily the antigens can be identified by the B cell receptor complex, that is immunoglobulins. But sometimes these antigens, which is opsonized by the complement proteins, we have discussed under the section of complement system that C3b is a very beautiful opsonizing molecule. Sometimes these antigens will be opsonized with the help of complement proteins like C3b, and this C3b bound antigen can be identified by the complement receptor which is present on the B cells. Okay, so the antigen can be identified by both the B cell receptor complex, and also sometimes if it is opsonized, it can be identified by the complement receptor also. Okay, and this entire process will give the signal 1 to the B lymphocyte. Okay, so this interaction of antigens with the B cell receptor complex and the complement receptor, this is going to give the signal 1. This is going to give the signal 1 to the B lymphocyte. Sometimes, if the B cells are activated with the help of T lymphocytes, okay, or macrophages, okay. So basically, the B lymphocyte can be activated in a T cell independent manner. It can be activated by a T cell dependent manner or T cell independent manner. Remember, T cell dependent manner activation of the B lymphocyte is very important, and this will cause more strong or more efficient B cell activation. But sometimes B cells can be activated without the help of T cells, either directly, or sometimes with the help of follicular dendritic cells, or sometimes maybe with the help of macrophages. Okay, but T cell dependent activation is very important. Now, if the B cell is activated with the help of T cells, now the T cells, they will have a molecule that is known as CD40 ligand. That is known as CD40 ligand. So when the T cells will activate the B cells, they will activate the B cell receptor complex, which will give the signal 1. But along with that, they will also activate the CD40 receptor with the help of CD40 ligand, which will provide the signal 2. And that will ultimately activate the B cells to convert into either a plasma cell. So this entire process of activation with the help of both signal 1 and signal 2, it will be converted to either a plasma cell or a memory B cell. It can be a memory B cell, or it can be converted to a plasma cell. And these plasma cells, they are going to produce the antibodies. They will produce the antibodies. These antibodies are directed against that antigen which has activated the B lymphocyte. Okay, so now this antibody can go and damage those antigens. Okay, so this is about the B cell receptors and what is the fate of B cells. We'll also discuss in detail about the T cell dependent and independent activation in subsequent lectures. Okay, so I'll just revise it again. So B cell receptor complex includes the immunoglobulin molecule along with the associated proteins. Immunoglobulin can be IgM or IgD. Associated proteins are immunoglobulin alpha and beta, otherwise known as CD79A, CD79B. The co-receptors include complement receptor, which is known as CR2 or CD21, which is also a receptor for Epstein-Barr virus. Another co-receptor is CD40. Okay, so whenever the antigen is identified by the B lymphocyte, it will be identified by the B cell receptor complex, and if it is bound to the C3b complement protein, it can also be identified by the co-receptor, co-complement receptor. And this B cell receptor complex, along with the co-receptor, along with the complement receptor activation, will give the signal 1. Okay, and the signal 2 can be given if the B cell is activated by the T lymphocyte. If the B cell activation is T cell dependent, the signal 2 can also be provided. The signal 2 is provided with the help of interaction between the CD40 ligand of T cells and the CD40 receptor of B cell. Both these signals will activate the B lymphocyte. Similarly, what we have discussed for T cells, similarly B cells also need both the signals for proper activation, and this will cause conversion of the B lymphocyte into either memory B cells or the plasma cells. Of course, there will be multiple intermediate steps like we have discussed when I discussed the B cell activation in the lymph nodes. I have told you whenever the B cells are activated, they will be converted to centroblasts, then centrocytes, then there will be some kind of selection process, and ultimately they will produce the plasma cells and the memory cells. The plasma cells will produce the antibodies which will go and act against the antigens which has primarily activated the B lymphocyte. Okay, so this is the concept. And we have discussed previously that T cells will also have two signals. The first signal is, the signal 1 is given by the T cell receptor complex proper, along with the CD8 molecule, along with CD8 or CD4 molecule, and the interaction of B7 with CD28 is going to give the signal 2 for the T lymphocytes. Okay. So now we have discussed about the T lymphocytes and the basics of B lymphocytes. Now we'll see some other cells which take part in the adaptive immunity. Next one is dendritic cell. It's a very important cell. Dendritic cell is component of both innate immunity and adaptive immunity. Now, dendritic cells are, these are prototype antigen presenting cell, or you can say the professional classical antigen presenting cells. There are actually 4 points you have to remember about dendritic cells. I mean, these 4 points will tell us why these are professional antigen presenting cells. The first one is the location. So they are basically present in the under the epithelia of skin. And also they're present in the interstitia of tissue. Interstitia of tissues. Okay, so basically they're located in such areas where the antigens can be easily identified, or you can say the first contact of antigens can be made. Okay, so the first thing is about the location. The second thing is receptor. Actually, these dendritic cells will have various types of receptors, like toll-like receptors or the lectin receptors, which can identify various products or various components of the infectious pathogens or the antigens. Okay, so they will have various receptors which can identify the microbes or the foreign antigens. The third point is, once they interact or once they engulf, or once they identify the antigens, they can take up the antigen and they can migrate into those areas where they have to interact with the T cells. Basically, the dendritic cells, they will process and express or present the peptide antigens to the T cells. Okay, that is the primary function of dendritic cells. The primary function is they will process the antigen, peptide antigen, and they will present the peptide antigen to the T lymphocytes with the help of MSC molecules. Because they have MHC molecules, of course, they will have both MSC class 1 class 2 molecules. As I have told you previously, class 1 molecule will be present in all the nucleated cells. So dendritic cells, they are going to have the class 1 molecule. And because they are antigen presenting cells, they will also have the class 2 molecule. Okay, so that means they can present antigen to both CD8 positive cytotoxic T lymphocytes and CD4 positive helper T lymphocytes. Okay, so basically they will present peptide antigen with the help of MHC molecule to the T lymphocytes. For that, they have to migrate from their original location to those areas where the T cells are dominant. That means once they take up the antigen, they will go to the spleen, periarteriolar lymphoid sheath. In the lymph node, they will go to the paracortex. I mean, we have already discussed that inside the spleen, the periarterial lymphoid sheath is the region where the T lymphocytes are present. And inside the lymph node, paracortex is the area where the T lymphocytes are present. So that means the dendritic cells, once it will take up the antigen, it will carry the antigen, process it, and it will migrate to those areas where the T cells are dominant, and there it is going to present these peptide antigens to the T lymphocytes. Okay, so these are the 4 properties which makes these dendritic cells the classical professional antigen presenting cells. Okay. Now, what are Langerhans cells? Langerhans cells are nothing but immature dendritic cells which are present in the epidermis of the skin. These are actually type of immature dendritic cells that are present in the epidermis of the skin. Okay. Now there is another type of dendritic cell that is named as follicular dendritic cell. Okay, so this is follicular dendritic cell. When I discussed about the lymph nodes, I have discussed about these follicular dendritic cells. The point you have to remember is the function of follicular dendritic cell is completely different from the dendritic cells. Okay, the function is completely different from the dendritic cell. The only similarity is their appearance. Okay, because they have these dendritic processes from their cytoplasm, these are known as dendritic cells. But the function of follicular dendritic cell is completely different from the dendritic cell. I have discussed in detail in the lymph node section. Okay, so basically they will have receptor. They will have FC receptor for the immunoglobulin molecule, especially the IgG. As I have told you previously, IgG can act as opsonizer. So it can actually opsonize the antigen. And if the IgG molecule has opsonized one antigen, that particular antigen can be identified by this follicular dendritic cell because they have receptor for this IgG molecule. Similarly, they will also have receptor for C3. They will also have receptor for C3b molecule. C3b also act as an opsonizer. Okay, so because they have this Fc receptor for IgG molecule, they can identify those antigens which are opsonized by IgG. They will also have receptor for C3b, which can identify it. Through this, it can identify those antigens which are opsonized by C3b. And these antigens can be expressed, can be presented to the B cells, especially in the lymph nodes. Okay, so these are presented to the B lymphocytes and it can activate the B lymphocyte. But more importantly, the predominant role of follicular dendritic cell is to identify those B lymphocytes which do not have receptor for these antigens. Okay, so if the B cells, if the B lymphocytes, if they express antigens, if they express the receptors for self antigen, that can be identified by this follicular dendritic cell, and it helps in the apoptosis of those B lymphocytes, those self-reactive B lymphocytes. Okay, and that happens in the germinal center of the lymph node. So all those concepts I have discussed in detail in the chapter of lymph node, so I'm not going to discuss all those things here again. So this is about the dendritic cells. Now we'll discuss about okay. I have forgotten to put the macrophages. So next one is about the macrophages. The next type of cell that we're going to discuss under this adaptive immunity is about the macrophages. Basically, macrophages, they actually help in both cell-mediated immunity and humoral immunity. If you see the cell-mediated immunity, the first function of this macrophage is it can act as antigen-presenting cell. Okay, so macrophages are also antigen-presenting cells. They can present antigens with the help of MHC molecule, especially MHC class 2. Of course, it can have both MHC class 1 and class 2. It can have both class 1 and class 2 because it is a nucleated cell. So it will have class 1. It is antigen presenting cell. So it can have class 2 molecule also. With the help of class 1 and class 2, it can present antigen to the T lymphocytes. It can present antigen to the T lymphocyte and help in activation of the T lymphocytes. Also, during the effector phase of cell-mediated immunity, I have told you previously also, when the cytotoxic T lymphocytes or the CD8 positive T lymphocytes are activated, they can produce certain cytokines which can ultimately activate the macrophages. It can activate the macrophages, and these macrophages help in the clearance of the antigen or the microbial pathogen. Similarly, the TH1 subtype of helper T cell, it can also activate the macrophages. Of course, this activation of macrophages happens with the help of a very important cytokine, that is interferon gamma. Okay, cytotoxic T lymphocytes, they can activate the macrophages. TH1 lineage of CD4 positive helper T cell can also activate the macrophages, and these macrophages helps in clearance of the antigens. Okay, these concepts I have discussed when I discussed about the hemophagocytic lymphohistiocytosis. And I have told you, if this process of macrophage activation cannot be controlled, that can lead to hemophagocytic lymphohistiocytosis, otherwise known as macrophage activation syndrome. Okay, so this is the role of macrophage in the cell-mediated immunity. They also have role in the humoral immunity. Basically, humoral immunity is mediated by the B lymphocytes and their products, that is antibodies. One of the important antibodies is immunoglobulin G, and I have already told you multiple times that immunoglobulin G is a very important opsonizer. So they can actually opsonize the antigens. They can bind to these antigens, and once they opsonize the antigens, these molecules can be identified by the macrophages, because macrophages also have this Fc receptor of immunoglobulin G. So if you see the immunoglobulin G molecule, it will have a FAB portion, FAB portion, that is the the antigen binding portion, and it also has a Fc portion, that is the constant region, Fc portion. This Fc portion can be identified by the Fc receptors, and these macrophages have this Fc receptor. Through this, it can identify those antigens which are opsonized by the immunoglobulin G. Okay, similarly, what I have told you for follicular dendritic cells. Okay, so various cells in our body, they have this Fc receptor for immunoglobulin G molecules. And through this Fc receptor, they can identify those antigens which are opsonized by IgG. And once they're identified, this macrophage can be activated, and this activated macrophage can actually phagocytose this particular antigen. Okay, so in the effector phase of both cell-mediated immunity and humoral immunity, macrophage is going to play a major role. Okay. Now we'll see the tissues of immune system. So this is just a you can say overview, because we have already discussed in these things in detail in separate lectures. So primary lymphoid organs include bone marrow, thymus. Secondary lymphoid organs include spleen, lymph node, and the cutaneous and mucosal lymphoid tissues. Among all these things, we have already discussed about the bone marrow, we have discussed about the thymus, we have discussed about the spleen, we have discussed about the lymph nodes. Now, little bit about the cutaneous and mucosal lymphoid tissues. So cutaneous lymphoid tissues, they are present under the epithelia of the skin, and mucosal lymphoid tissues, these are present in the epithelia of GIT and respiratory tract. The lymphoid tissues that are there in the GIT, two important lymphoid tissues include the tonsil, the tonsils, the palatine tonsil, and the Peyer's patches. Tonsils and Peyer's patches, these are types of mucosal associated lymphoid tissues. Okay, and these are all secondary lymphoid, spleen, lymph node, and this mucosal associated lymphoid tissue. These are secondary lymphoid organs. Bone marrow and thymus are the primary lymphoid organs. Okay, so this is an overview of all the cells that are involved in the adaptive immunity and the tissues or the lymphoid tissues that are involved in this process. Now, the last thing that I want to discuss is about the lymphocyte recirculation. Now, whatever we have studied till now, we'll just have a recap of all those things. The first thing is we have to start from the primary lymphoid organs. As I have told you, primary lymphoid organ include the bone marrow and the thymus. The B cells, they are produced and matured. They're produced and matured. I mean, developed inside the bone marrow itself. Okay, so they are produced and developed inside the bone marrow itself. From the bone marrow, they can go into the peripheral circulation. From the bone marrow, they can go into the peripheral circulation, and from there, it can populate the secondary lymphoid organs. And we have already discussed inside the spleen, they are present. I mean, inside the spleen, they are present in the white pulp. Okay, and just near the central artery. And inside the lymph node, they are present in the cortex as lymphoid follicles. In spleen also, they will be present as lymphoid follicles. Okay, and also they will be there inside the mucosal associated lymphoid tissues. But what happens to the T cells? So the T cells, they'll be produced inside the bone marrow. However, the T cells have to come to the thymus for further development. So T cells will come to the thymus for further development. From the thymus, once they are developed, once they're matured, they will come to the circulation. And now they'll go and populate the secondary lymphoid organs. We have discussed that inside the spleen, they are present as peri-arteriolar lymphoid sheath. And inside the lymph node, they are present in the paracortex. They are present in the paracortex. And also they will be present in the mucosal associated lymphoid tissues. However, all these lymphocytes, these are naive. That means these are immunologically inexperienced. They have not exposed to the antigens. They'll be exposed to the antigens predominantly in these secondary lymphoid organs. Okay, so these are all secondary lymphoid organs, and these naive B and T lymphocytes, they'll be exposed to antigens in these secondary lymphoid organs with the help of, I have told you, with the help of, especially with the help of the antigen presenting cell, that is the dendritic cells. Okay, and once they are activated, once they are exposed to antigens and activated, the B lymphocytes, they will produce plasma cells. Okay, so basically the B lymphocytes, they will produce the plasma cells, and these plasma cells, they are going to produce antibodies, and these antibodies will go to the site of infection. It will go to the site of infection to mount immune response against the antigen or against the microbe. Okay, so these antibodies, they will go to the site of infection. But what happens when the T lymphocytes are activated? When they are activated, they will come to the circulation as activated T lymphocytes. These are all activated T lymphocytes. And these activated T lymphocytes, as such, they will go to the site of infection. They will go to the site of infection. Okay, so there is a small difference between the B lymphocytes function and the T lymphocyte function. You can understand from this diagram that the T lymphocytes, when they are activated, they are known as activated T lymphocytes, and as such, they will go to the site of infection and they will have their function. They will perform their function. However, the B lymphocytes, they are not going to leave the lymphoid organs. The B lymphocytes, when they are activated, they will produce plasma cells. These plasma cells, they will produce antibodies which will go to the site of infection. So B cells and plasma cells, they are not going to go to the site of infection. They'll just produce the antibody molecules which are going to have their function in the site of infection. Okay, so this is the difference between the function of ultimate function of T lymphocyte and B lymphocyte. Of course, some of these plasma cells, they will go into the bone marrow. They'll go into the bone marrow. Usually, plasma cells are not found in the peripheral circulation. This is just to, just to simplify the things. Actually, the plasma cells, they are found inside the secondary lymphoid organs. From there, they will go to the bone marrow. But usually, in the peripheral circulation, it is difficult to find the plasma cells. The thing that you're going to find is antibodies. Okay. So this is about the lymphocyte recirculation. So all these lymphocytes, they are not static at one location. They will be circulating throughout the body. They will be populating various lymph nodes. They will be going to the spleen, and they'll be constantly moving inside the circulation and exposed to antigens and produce their response. Okay, so this is about the adaptive immunity in a nutshell. More details of this we'll discuss in the subsequent lectures.